So the first talk will be by Serena Nik Zainal. She's a clinician, a scientist at Cancer Research UK. She's also a consultant in clinical genetics. Actually, I think she's obviously a fantastic scientist, but she's still doing some clinical work, and she enjoys music. That's what I could find on Google. So today, Serena will tell us about her work in harnessing the value of whole genome sequencing in human cancer.
Hang on, there we are, unmuted. Thank you very much for that kind introduction. I must look this up on Google. I'm not quite sure what that's all about, but yes. Okay, so you can see my screen, I hope.
Yes.
Great. Okay, so I hope you can see my full slide. So thank you again for the invitation to come and speak today. These are my disclosures. And for today, what I'm going to do is talk about an area of particular value that comes from the ability to sequence the entire genome or whole genome sequencing, an area called mutational signatures. I'm sure a lot of you have heard about it already. I'm going to focus particularly on how we use mutational signatures to generate clinical applications. I'll touch a little bit on some of the experimental work that's been done to validate some of these otherwise, abstract analytical patterns. And then I'll finish off with a story about the impact of whole genome sequencing and mutational signatures on a patient. And so this signatures field isn't new, but I will just give a little bit of an overview. So I apologize if you already know a lot about this, but as a cell turns from being a normal cell to being a cancer cell, it doesn't just acquire the handful of driver mutations that people have focused on for decades in cancer research. It also acquires a large number of passenger mutations that arise because of the mutagenic processes that are occurring as that cell turns from being a normal cell to a cancer cell. Now, some mutational processes may be perfectly normal. They're happening in all your normal cells, for example, replication errors. Some may be due to environmental agents that damage DNA of exposed cells, for example, tobacco smoke on lung DNA. Some mutation processes may be endogenous and intermittent, or the continuous results of defective DNA repair pathways, for example. But each of these processes will leave a characteristic imprint, a signature on the genome. But when you sequence a cancer genome, what you get is a final portrait. It's a composite of all those signatures added together. And it may look like a random collection of mutations. But in the past, it's been shown that if you had genome sequences from multiple tumors of the same tumor type, it's possible to use mathematical methods to extract mutational signatures, i.e. identify these rectangles, and then even quantify the amount of the signatures, which is to quantify the size of these arrows. So historically, the mutational signatures field started with substitution, single base changes. That's simply because it was the easiest class of mutation to call when we first started doing whole genome sequencing. No other reason apart from that. Okay, so you can do mutational signatures in any mutation class, but a lot of the work's been done in substitutions. So historically, we presented it like this. You have the 6 main classes, and you take the 5' and 3' sequence context into account. There's 4 possibilities, 5' and 3', so that's 4 by 4 is 16. 16 multiplied by 6 gives you 96 channels. The signature is the entire 96 channel pattern. It may have certain dominant characteristics. So here, the tallest bars are at CPGs. And this signature is due to deamination of methylcytosine. It's seen in every single tissue type. It's seen in normal tissues. It's a perfectly normal signature that's happening in all our cells. There are a number of other intrinsic or endogenous mutational processes. For example, the APOBEC ones also deamination, which because of differing repair pathways that try to fix that APOBEC deamination can result in multiple different signatures. And here are a couple of examples of signatures due to environmental mutagens, very well-known ones. Signature 7 with these dominant C to T mutations is due to UV light seen in skin cancers. Signature 4, this dominant C to A pattern is seen in lung cancers due to tobacco smoke. And this T to A pattern is classic of aristolactic acid in liver and kidney cancers of people who've ingested aristolactic acid. I stress to add that a lot of these patterns were actually reported well before whole genome sequencing started. It was reported in the 1990s by people who worked on reporter assays in HBRT and P53, It's just that now that you've got the power of whole genome sequencing, you can see just the scale of it, the amount of it, and you can get more granularity by presenting it in this 96 channel pattern. So what does it look like in individual patients? So here I'm going to show you 4 different tumor types: breast, cervix, a kind of leukemia, and lung cancers. Each bar represents a patient. You can see that they all have different numbers of total mutations, and they have different colors representing the different signatures. So the arrows are pointing at the green signature, signature 1, that's deamination of methylcytosine. Every sample has a bit of it because it's common and it's normal. Then this is ApoBec, this is the pink stuff. A lot of samples have it, but not every tumor type has it. But some tumors have got a huge number of it, a hypermutator phenotype, even for an intrinsic mutational process. And that's usually an indicator of something else that's wrong with a tumor, where there's replication stress usually. And then the arrows are pointing out the blue signature, signature 4, which is tobacco smoke. And you're only seeing it in lung cancer, not the other tumor types. But note the mutation rate, right? So environmental mutagenesis tends to have really huge mutation rates. So that's just to give you a flavor of what signatures are, what they look like in individual patients. But I would stress that the field is still early. It's still young. It's still developing. The number of mutational signatures in the literature is not static. This is changing. It has changed even in the last 8 years. It's going to change again very shortly. The classes of mutational signatures is also increasing. We now have double subs and indels and rearrangements, so nothing is gospel. And the third thing to say is that the number of mutational signatures in a data set, when you try to identify the number of signatures, is actually subject to some uncertainty. When you try to factorize or decompose a complex data set, this is a typical problem that you can have multiple potential solutions. And the best way to identify the true number of signatures remains a little bit difficult to resolve. So you could be one signature out, or too many or too few. But in general, the main signatures, the important ones, tend to just be there, whether you go for 4 or 5 signatures, we're one out. Generally, the big important ones are pervasive. Now, the next thing to note is that the mutational signatures do not all behave in the same way. Some are subject to more uncertainty than others. So this is a PCA plot of attempts to identify signatures from a complex data set. You're just doing it again and again and again and again and again. And you can see, for example, this arrow is pointing to a signature that's very clear. It's got very tall, beautiful peaks. It's very distinctive and easy to extract. So it's always extracted in the same way. It forms a very discretized cluster. By contrast, these signatures form this ring because they can sometimes be misclassified as each other. And that's kind of important to note, because one of these signatures is one of the ones that we highlighted in the past as signature 3 as being a feature of BRCA-ness, something that was seen in BRCA1 and BRCA2 tumors. And people use this signature to try to classify tumors as to whether these are patients that could be sensitive to some of the drugs that have been created for BRCA1 and BRCA2 deficient tumors. So that's PARP inhibitors. But as you can see, it can be a little bit difficult to be certain whether you've got that signature in your sample. And so there is some uncertainty here, and there is some danger in erroneously classifying your tumor based on a single signature. So I think the bottom line is we need to be a little bit cautious in making a clinical judgment on a tumor sample based on a single mutational signature. And so it's not a perfect science at all. There are some caveats. I mean, I'm somebody who really wants signatures to be used clinically, but I think it's important for us to just be aware of some of these caveats. So going into clinical applications then, I've just said that using a single signature is probably a little bit brave. And so what we do try to do is try to look for ways to use mutational signatures in order to stratify patients, but to try to make it as robust as possible. And what I'm going to do now is contrast 2 whole genome sequence breast cancers from 2 patients for you to see for yourself, actually, that some of these signatures are so clear, you can use them to apply machine learning methods in order to create algorithmic predictors. So here's a ER positive breast cancer that is the sort of tumor that would usually be cured. And there's all her whole genome data. In fact, she's got a total number of 2,000 substitutions, mainly signatures 1 and 5, which doesn't matter, but they're just kind of benign, boring signatures. And this sample instead has a BRCA1 germline mutation with loss of the other allele. So this is a BRCA1 deficient cancer. And just looking at this circle plot, you can see that it looks very different. But she's got very different substitution signatures, signatures 3 and 8 that I mentioned earlier. She's also got deletions with microhomology at the junction, first reported by Maria Jason, and it's indicative of a BRCAness. She's also got lots of rearrangements, this lines going throughout the center of the genome, 300 of them beautifully dispersed through the genome, a classical feature of BRCA1 deficient tumors, and a lot of copy number losses throughout the genome. This pink stuff are whole chromosomal, whole arm losses going throughout the genome. That is the feature that's used by current tests. The Myriad score is based on these copy number losses. But the point is that all of these features together, not one, all of these different signatures together can now be used to inform your patient stratification. And so we apply machine learning methods on these two groups of patients, ones that have the ER positive tumors and the ones that have the BRCA1 or BRCA2 deficiency. And we created this algorithm called HR detect. So a high HR detect score predicts BRCAness and a low score predicts not. And you've got a lovely sigmoidal curve here. It's nice and clear, and we've taken a cutoff of 0.7. But to our surprise, the number of samples that have high scores was rather large. It was 22% of the cohort, 124 of 560 whole genome sequence breast cancers. So that's a lot more than we thought we should find. We thought, maybe 5%, 10%, but not 22%. So we looked at this 22% of cases in more detail. And we had 22 positive controls. So we knew they had germline mutations in BRCA1 and BRCA2. So we knew we should find them, and we did. But we found an additional 33 cases that had BRCA1 and BRCA2 germline variants that did not know that they had them in the past. So either they had small families, or they did have a family history of BRCA1 of breast and ovarian cancer, but they had limited genetic testing and didn't look at the whole gene, because 15 years ago we didn't. And so we missed some of those. And then there were 22 women who had somatic mutations in BRCA1 or BRCA2, or promoter hypermethylation of BRCA1. This one black sliver is actually one of the 33, so that's 32 here. And this one has got a germline mutation, but the other allele is still intact. But in about a third of cases, we couldn't find a cause. So we can see the patterns. It looks like it's BRCAness. It looks classic of BRCAness, but we cannot find a germline or a somatic or promoter hypermethylation of BRCA1 or BRCA2. And we looked for some of the other genes as well involved in the HR pathway. We just couldn't find a genetic or epigenetic driver. But that's kind of important, right? Because if you're going to use mutational signatures to try to stratify your cases, I mean, this is going to be a cohort of patients that you're going to miss if you just did targeted sequencing. So some people will say, don't whole genome sequencing, it's too expensive, just sequence the genes, but you're going to miss this 1/3 of cases. Okay, so at this point, we've got an algorithm. It can predict biology. That's great, but does it predict outcome? Is it clinically useful at all? And so I had the good fortune of collaborating with colleagues in Sweden, UN staff in Åkerborg, and they had started the SCAN-B project from 2010, which basically said, let's just recruit every single breast cancer patient that walks in through the door in the whole of the south of Sweden. So these are all names of district hospitals in Sweden. And it was without exception. Every woman was invited to join this project. This was a standard, routine molecular diagnostic pathway. There was nothing special about it. These patients would come in, they would get recruited, they would get a sample put into bank, and then a sample used for clinical diagnostics. And so they had 12,000 patients, which I can't afford to sequence. So we said, let's just do an area of unmet clinical need, because a lot of breast cancers are cured. Let's focus on triple negative breast cancer. And so we sequenced all the triple negative breast cancers between 2010 to 2015. And the wonderful thing about the Northern Europeans is that they have fantastic treatment and outcome data. So these are all patients who've had standard of care for triple negative breast cancer in the south of Sweden. So the first thing to say is that more than half the patients had HR deficient tumors, so 58.6% had high scores. And that might sound like a lot, but it's what we expected, because triple negative breast cancers tend to have a lot of BRCAness. We don't necessarily know the cause for their BRCAness, and we don't necessarily know who are the ones who are going to do well and not do well, but we do know that there's quite a lot of BRCAness in TNBC. Okay, so importantly, does it prognosticate? Does our algorithm prognosticate? So we've got here Kaplan-Meier plot. In black are all the patients with high scores, and in gray are all the patients with low scores. And as you can see, the patients with high scores are the ones that do well on standard of care. But we did not have the genetic or epigenetic cause for some of these cases, right? We had it for about 2/3, and we was missing in about 1/3. So when we look at this cohort in more detail, we split them up into the patients where we could find a genetic or epigenetic cause in black versus the ones in gray where we couldn't. We found that there was no difference between them, meaning that the mutational signature approach, the algorithm itself, was able to prognosticate on who was going to do well or not do well, based on, or using HR Detect based on standard of care for triple negative breast cancer. And again, this is important, right? Because there's 1/3 of this cohort that we cannot find a genetic or epigenetic driver. And for this cohort, we had everything. We had methylation, we had transcriptomics, we had the genomics. I would say that we did find some additional genes. So we found that you had to be biallelically mutated for PALB2, or you'd have to have RAD51C hypermethylation, and that would produce a similar BRCA kind of profile. In fact, it was very similar to BRCA2. So the bottom line is, taking this multiple mutational signature approach through HR Detect, we're able to discriminate tumors that have improved clinical responses to triple negative breast cancer therapy, demonstrating the utility of the signature-based tool. So I'm going to move on to just high-level stuff here on some of the experimental work that's gone on to try to support some of this stuff, because a lot of it is, abstract analysis. Okay, we've put some into some clinical studies. We've now gone into a proof of principle clinical trial with Clovis, but what is the experimental evidence that these signatures really are what they are? And so we have done a piece of work where we've used a human IPS line. It's isogenic, and we've either treated them with environmental mutagens, the sort of known and suspected IARC carcinogens group 1 and 2, and then we've done CRISPR-Cas9 knockouts of different DNA repair replication genes. Now, I'm a clinical geneticist. I deal with children with rare genetic disorders and learning disabilities. And so I've recruited patients who have recessive DNA repair defects through this project called Insignia. And so I've got a collection of lines that have their normal lines. These are not cancer lines. These are normal stem cell lines. And yeah, we're looking at mutational signatures to see whether we can experimentally recapitulate what we're seeing in human cancers. And the bottom line is the answer is yes, we can. We also find some new things. I don't have enough time to go into all of the details. I will focus on the CRISPR-Cas9 knockouts. I will say that we have published on the environmental mutagens in 2019, but today I'm going to focus on some of this stuff, not even all of it in the interest of time, just in one particular area to demonstrate some of the mutational mechanistic insights that we can get from it. So this was work I did with Joe Giritny in Zurich and Bill Scarans at the JAX. So we knocked out all of these different genes in all of these different repair pathways. We didn't manage to knock everything out. It was all semi-high throughput. So of course, there are going to be some genes that are going to be impossible to knock out, but this is what we managed to get. I'm going to focus on mismatch repair, because mismatch repair deficiency tends to be a dumping ground of high tumor mutational burden and microsatellite instability. And I hope that you'll see that actually there's a lot more granularity than that. So we knocked out all of those genes. And the first thing to say is that there are quantitative differences in substitution and indel burdens between the different genes. The next thing to say is that there are qualitative differences as well. So the top three are knockouts of MSH2, MSH6, and MLH1, and they are nearly identical to each other. It's pretty extraordinary, but they are really quite different to the knockout of PMS2. Now, one of the things was, is this really real, or is this just part of your, something to do with your line, your cell line, your IPS line? And so what we did was we looked at patients who had constitutional mismatch repair deficiency. So these are kids who have inherited 2 mutations from Lynch syndrome parents. So they are recessive. They've got both of their mismatch repair, inherited mismatch repair genes knocked out. These are kids who usually get these cutaneous manifestations, cafe au lait patches, and they tend to get pediatric malignancies and often die within the first decade of life. So we made iPS cells from them, and we did the whole genome sequencing. And what you're going to see is here we had 2 patients with PMS2 mutations, and 2 of them with, actually it's MSH6. I don't know why this says MLH1. I do apologize. It's MSH6. And then on this axis, we have the patterns from the knockout genes. And what we're looking at is whether there are they are similar to what we're seeing in the knockouts? And the answer is yes, they are. So these mutated ones are very similar to the MLH1, MSH2, MSH6 profiles, whereas the PMS2 mutants are very similar to the PMS2 knockouts. So that gene specificity that we saw in the IPS lines are also seen in cells that are derived directly from patients. Furthermore, that gene specificity is also seen in cancers. If you look hard enough, most of the time we just say it's mismatch repair deficient. But in fact, the MLH1 null tumors tend to have very particular substitution signatures, and they have this particular indel pattern, which we all recognize as microsatellite instability. These are deletions at polynucleotide repeat tracts. And then the PMS2 mutants have these green mutations. These are signature 26, which is a particular substitution pattern, and they actually have a different indel profile. They mainly produce insertions rather than deletions. These are still polynucleotide repeat tracts, but it's kind of curious that they produce insertions more than deletions instead in the PMS2s. So, so we sort of demonstrate that there's a gene specificity. There are differences quantitatively and qualitatively. One of the other things we did then was to try to figure out whether we could understand what the sources of damage were that resulted in this mismatch repair deficiency signatures. Now, in the interest of time, I don't have enough time to go into all of the details. I will tell you quickly that the C2A component we think comes from guanine oxidation, and the C2T and T2C components come from misincorporation by polymerase alpha, delta, epsilon errors, which kind of makes sense. But what I want to draw your attention to is this little T2A peak here, which is tiny, but you can see that it's really consistent across all of the signatures. It's modest, but very clear. And when you look at it in more detail, you look at the bases a bit further downstream and upstream of the T2A change, you find that it's sitting at where poly A's and poly T's are abutting each other. So there's this excess of T2A transversions occurring when poly A's and poly T's are next to each other. And because polynucleotide repeat tracts predispose to indels due to replication slippage, a known source of mutagenesis in mismatch repair deficient cells, we hypothesized that these T2A transversions are the result of a mechanism called reverse template slippage. This was Joe Jaricni's term. So let me try to walk you through it. Under the usual circumstances, when mismatch repair is intact, we should have error-free synthesis. So here's a poly A and a poly T tract. However, a polymerase replicating across a mixed repeat sequence such as this could cause a little insertion deletion loop, this A sticking out here. And then you'd get an incorporation of 5 instead of 6 T's opposite this, what should have been 6 A's. And now if at this point, the template reverses back to its original alignment, so the IDL then disappears, what you're going to get is an AA mismatch. And that would result in the T2A transversion in one of the daughter cells in the next round of replication. Now, if the slippage remained, which is what we usually expect, then you get the standard phenotype of the single nucleotide deletion, a feature that we know to be seen in mismatch repair deficient cells. By having the experimental data and by looking at the information in great detail, you can sometimes try to figure out some of the mechanistic insights that have resulted in some of these signatures. So in my last couple of minutes, I'm just going to finish with a case, because it's very close to my heart, why we work on the whole genome data and why we use mutational signatures in great detail, but also that you have to interpret it and not just produce it from an algorithm and go, you know, this is what it shows. You do have to think about the data a little bit. So this is Joshua Barnfather. I have his consent to talk about him. He's 30 years old. We actually know him from our DNA repair clinic. He's got xeroderma pigmentosum type C. So XP genes are involved in nucleotide excision repair, and they usually fix UV damage, but kids with XP cannot fix UV damage. So they get an excess of lentigines, and they get a horrible increased risk of skin cancer and terrible sunburn. And in the past, these kids used to die in their first, second decade of life. These days, they get meticulous photoprotection and they live into their adulthood. So Josh presents then in 2017 with this violaceous tumor in the medial aspect of his eyebrow, and it turns out to be something called an angiosarcoma, which is a rare cancer of the blood vessel lining. So he has a wide local excision and a skin graft, but within a few months comes back with mets in his lymph nodes, and then he has progression to his liver and pleura in spite of treatment for angiosarcoma. He just isn't responding at all. And by July 2018, this man is dying. I do his whole genome sequence, and we get this huge amounts of red dots, as you can see. And this looks like malignant melanoma. This is UV damage. Now, his drivers are boring, CDKN2A and P53. He doesn't have the drivers that are typically associated with angiosarcoma. When we look at his mutations, we see the classic pattern of UV damage that is typical of UV. Now, if you run this past my team right now, they're going to go, oh, this is like a malignant melanoma. But remember, he's an XP mutant. So you have to interpret all that data together and say, actually, for an XP mutant, this is probably normal. He cannot fix UV damage. So if I could sequence all of his normal skin cells, I'd probably find a huge amount of UV damage. What's really killing this man is this signature historically caused by mutations in the proofreading domain of polymerase epsilon. Now, initially, we couldn't find a mutation in polymerase epsilon. We could just see the signature. Eventually, through a lot of physical digging, we found a mutation present in a tiny subclone. So it's not in the major clone of his cancer. It's in a minor subclone. But I think that is what has gone off and seeded off all the nets and is what is killing this man. So Pol E mutated tumors are important to identify because they tend to be sensitive to checkpoint inhibitors. And so we did PD-L1 staining. Initially on the primary tumor, it was negative. And I said, do it on the Mets. And when we did that, it came up as positive. And so we now had the potential evidence to suggest that his tumor might be sensitive to checkpoint inhibition. Now, he had to raise his own money because in the UK, this is not yet licensed for use in metastatic angiosarcoma. But I can say that he got the treatment. This was him pre-treatment, and 3 cycles in, everything is starting to melt away. By 7 cycles in, it had all gone away. He's now finished all his treatment as of November last year. And his last scans were very, very positive. So it's been a couple of years. He's done really well. And it's a lovely story to end on. But really, the bottom line is that when you use whole genome sequencing and you use mutational signatures, there is some added value. But you do have to interpret all of that data together to get the best outcomes for your patients. So that's me. This is my acknowledgement slide. I'd like to acknowledge the leadership of a lot of the whole genome sequencing consortia, ICGC, BASIS, Hartwig, without whom we wouldn't have the data that I have access to in order to do all of my analyses, major collaborators, all of these people, and this is my team in Cambridge. Thank you very much. I'm happy to take any questions. Thank you.